Hi, I'm Madison Young.
I'm Alex Kimpel.
I'm Morgan Lyons.
I'm Cohen Solal.
And we are going to be teaching you about functionally redundant genes, or not, today.
Alright, so a gene is said to be functionally redundant if that gene codes for a function that's already being coded by another gene. And a good example of this is amino acids, and that's because there are only 20 different amino acid chains that exist, which you guys probably learned about in Bio1. But there are 64 unique codons, and a codon is a nucleotide sequence of 3 nucleotides, and that's like the whole U, C, G, and A thing that you guys learned about. And these 64 unique codons all create the 20 different amino acid chains. Just being that 64 is bigger than 20, that means there's obviously duplicates, but this acts as a safety net if there is a damage or a genetic mutation in one of the codons. And as you can see here in this slide, this is just a visualization of the last example. On the left picture, you can see all 64 codons, and that's, again, like I mentioned, the whole U, C, A, and G thing, and each combination of those 3 is a nucleotide. And there's 64 different combinations of 3. And then on the right, you can see the 20 different amino acid chains, and those are all made up by the codons seen in the left picture. So there's obviously the overlap, as I mentioned. With this definition I just gave you guys, there's some gaps in it, and you might have some questions yourself, because there's certain scenarios where it's kind of a questionable idea, and some examples of that. So if 2 genes have the same product, but go around it in different methods, are they considered functionally redundant? And then another example of a gap that this leaves is that if a gene has multiple functions, and one of those functions is seemingly redundant, but the other one does something that is unique to the organism, does that make the entire gene functionally redundant in the biological sense? And these questions are largely unanswered. There's no set ultimate definition for functionally redundant, so a lot of these questions that have these specific scenarios aren't exactly answered. So this gets into our presentation. Now that you guys know what functionally redundant genes are, the ultimate question is, are there such things as purely redundant genes, or does every gene have a function that makes it unique? And this sort of goes along with those questions on the last slide, so we'll kind of go into that during this presentation.
To answer some of these questions, scientists at the University of Utah performed multiple experiments on common laboratory mice with specific genetic mutations, such as the HOXB1 and HOXA1 genes. These genetic mutations, when studied under traditional developmental techniques, originally seemed useless and functionally redundant. They were thought to cause no effect on the mice. A total of 176 mice were used in the experiment, 8 to 10 males in each territory and 18 to 20 females in each territory. This was a total of 58 males and 118 females. Half of the mice in each mouse barn had 2 copies of the normal HOXB1 gene and half had 2 copies of the swapped gene with HOXA1. The mice were tracked for 25 weeks in the barns. The average survival for HOXB1A1 group is 189.5862 days, and the average survival for the control is 194.4773 days. The average territories occupied for the control was 4.758065. The average territories occupied for the HOXB1A1 group was 4.758065. The results for this experiment showed that the control group on average lived longer than the HOXB1A1 group. The average territories occupied for both groups was the same. 10 to 15% of the time, organisms can show normal phenotypes even when there are changes made to affect the genotype. It used to be thought that redundant genes had no use and were only used as a backup, but this experiment helped prove that knockout genes do, in fact, have a purpose as the genotype was altered.
Another experiment done to test for functionally redundant genes was done by the University of Florida. They tested the ethylene response in tomato plants. Ethylene is a plant hormone that aids in fruit ripening, abscission, seed germination, and stem elongation. It's also responsible for the triple response. In tomato plants, there are 3 of the same gene, arbutoposis EIN3, which helps in ethylene signal transduction pathways. To test for functional redundancy, one of the genes was taken out so that only 2 of the same genes were remaining. When this happened, the sensitivity to ethylene was reduced and there was not nearly as much growth as with all 3 genes present. This shows that even though there are 3 of the same genes, redundant genes, they all serve a purpose in plant development and are therefore functionally redundant genes. This next slide shows the results of the experiment. The Columbia is the wild-type plant. The next sample down is the tomato plant treated with the arbidipose EIN3 gene. The next 6 samples show when 1 gene was taken out, so only 2 remained. There are 6 different combinations of these. The conclusions that can be drawn from this experiment are that with the redundant genes taken out, the plants did not grow nearly as much as even the wild type, which supports the evidence from the mice experiment that there are in fact functionally redundant genes.
The conclusion of this experiment and this project. So, genes that are redundant can have different roles. Redundancy can be achieved by duplicate of a structural and a regulatory gene, and it is common in species in small population with large genomes for which their natural process is dominated by genetic drift. The meaning of it is the redundant genes are involved in parallel pathways. Various genes are functionally redundant and can back up disabled genes which can positively affect the life of the organisms. Our take-home message. 1. By looking at the results from the mice experiment and the tomato experiment, it can be determined that redundant genes do in fact have a specific function in some cases. Second take-home message is the natural selection cannot prevent the gene redundancy destruction due to debilitating mutations. And our third and last take-home message is the genetic redundancy is the key to understanding the robustness of organisms and their flexibility to rapidly adapt to different environments.